Respected viewers, I welcome you on this PIMA webinar series number 5. And today we have a very important topic on COVID-19, which is about the virus itself. What is the structure and testing of virus in terms of PCR and other antigen structures, and then the serology and antibodies of this virus. A lot of questions, not only in the layman's mind, but even the physicians always ask about the tests, its various methods, its indication, its timing and significance. And we have two very eminent experts in the clinical and pathology field. First of all, I'll introduce my dear friend for almost half a century, Professor Dr. Waheed Zaman Tariq. He is the very earlier virologist of the country. He did FPS and then FRC path and higher diplomas in bacteriology in UK. Served Pakistan Army, retired as brigadier, and then served very top-notch hospital of UAE, Tawam Hospital of Abu Dhabi, for about 10 years. And he has been serving recently here in Pakistan as advisor to the government of Punjab on COVID pandemic. I will request Dr. Waheed Zaman Tariq to start his presentation on PCR and antigens of COVID-19, which is SARS-CoV-2. Dr. Waheed Zaman.
Bismillahirrahmanirrahim. Thank you very much, sir. It is my pleasure and it is my great honor that I am with you. So this is one of the coronaviruses, and we started our work much earlier, maybe in January this year. Started delivering the lectures at different places and making the people aware of this virus. And having said this thing, I'll not go into detail, but initially the main thing was how to get the test reagents. It was virus was known early in January, and it was actually its genome was known on 24th of January, and by February we got the kits. And initially the problem was how to get them. So the world was stunned with that pandemic, and even in America, I can just show you the cartoons that America leads in the world in the COVID testing, just White House staff. So that is how the things were initially. But as far as the virus is concerned, it can be seen by electron microscope, but electron microscope does not tell you which coronavirus it is. There are other 6-7 coronavirus which are affecting the humankind. It is very difficult and sturdy method, and we don't have many electron microscopes available. Viral isolation is again a difficult process. Animal inoculation we don't do anymore. And by virus isolation, not only the answer comes in 10 days, but it is not that sensitive method, I can say. We are left with the viral components, and viral components, the virus is made of the proteins and RNAs. And proteins, sometimes we detect the antigen of certain viruses. And RNA, which is the viral genome, it is about 30 kilo Dalton, and that is about 28, 29 genes are there in this virus. And this RNA now can be converted to DNA, and we can pick it by the PCR. Gene sequencing is just done in research labs, and that is just for the purpose of knowing the different strains and different epidemiological purposes. So the important thing is the test starts with the taking of specimen, and specimen is taken by the person who is saving himself first and environments. And the swab, I always tell, is not the cotton swab, it is the Teflon coated. And then we put in the nose and we touch the nasopharynx, and we take it. And then important thing is the medium where we put this swab, and then the transportation is very important, and in time testing is very important. So that is where we start. And antigen testing is not that popular in this virus. I'll let you know how it is. So the viral test, as it has been just told, that it is a serological test. I'll not touch that. And then the viral RNA, this is the virus, outside is the protein, these are the spikes, and inside is RNA. RNA is extracted out of the virus, and then by the process of reverse transcription, we convert the RNA into DNA copies, and that DNA can be tested by PCR, and or the sequencing is done. So there are different methods. I will not go here in that detail, but different diagnostic tests are available at the moment, and I just base on the direct testing and leave the antibody testing to my next speaker. So this is the virus, which is about 120 nanometer in diameter. And as I have mentioned, inside is the RNA, which is about 30 kilodalton. And then there is spike protein, S proteins, and membrane protein, and then nucleocapsid protein, which just covers this RNA. So just if you remember, S protein, M protein, and nucleocapsid protein. And as the virus carries its own genes, so there is some RNA dependent RNA polymerase, because human body doesn't have RNA dependent RNA polymerase. So four antigens, which I will just let you know. Nucleocapsid, I have just mentioned this protein, which is on this RNA, and then spike proteins. These spikes actually attach to the ACE receptor, which is there in the respiratory tract and other cells, and then envelope protein, which just keeps the RNA protected from the environment. And then M protein, and this is the RNA. If you know the structure of the virus, I don't go in that detail, the same RNA is better represented. Spike proteins and M proteins. And then, well, if the person is infected here, the person will get this disease after few days. It is the incubation period first, which is 3 to 11 days, 3 to 11.5 days, and actually average is 5.5 days when person gets the high level of infection. And that is the time when the PCR is positive. So this is the viral, and that virus can remain for quite extended period in certain cases. I will just let you know, although person is no more infectious, but RNA can still be detected for 4 or 5 weeks in some cases, even more than that. But most of the people, the RNA clears anytime within 10 days of infections. So this is the symptoms. This is the blue area, and this is the viral RNA, which is detectable. And then the antibodies appear. IgM, we don't lay much emphasis on that, but this is the antibody which then goes on. So having said this thing, molecular testing is done on the secretions which we usually collect from the nasopharynx. But one thing is very important. I will just let you know, this test is not that very sensitive, because only 72 to 80% of the specimens which are taken from nasopharynx from the positive cases may detect the infection, and we still miss 20% or 25%. And why? Because the virus may be there in the nasopharynx, or it might not be there at that time. If you take nasopharyngeal aspirate, your yield is better. And if you take tracheal aspirate, then you pick in 92% of the cases. So taking the tracheal aspirate, as you know, is an invasive procedure, and the person who takes the specimen is exposed to the virus. So we interplay between that. Rather than taking the tracheal aspirate, we depend on the nasopharyngeal the swabs or aspirate, and then we can take two specimens to have more yield. And sometimes people take one specimen from nose and from one from throat, and they put on the same tube for that. All I told you about RNA, this is genome, and this genome, if you see, it is about 30 kilodalton, and this is the first gene, ORF1, this open reading frame 1 and 2, which, and then the RNA dependent RNA polymerase, and then the other S and N and E proteins are there. I don't go into that detail. So how we pick the RNA? As I told you that in the specimen, we first extract the RNA. Don't go into that detail. And then we put the probe in that. After extraction, the RNA is converted into DNA. And what is the probe? Probe is actually the small component of the virus, which is labeled with fluorescein dye. And if I will just go in a bit detail, I've already told you, tracheal aspirate with maximum yield, this with much less yield, but this is an invasive procedure. Transport medium is as important, swab is as important, and transport medium, I'm telling you that we made our own, and it just keeps the swab wet, and it doesn't, it avoids the contamination and things like this. We extract the RNA to have the genetic material, and then by reverse transcription, we convert into DNA, as I have said. So some labs initially went for pooling, and they started putting 10 specimens. And then if the pool was positive, then they were doing individual testing. I opposed it. Why I opposed it? Because it not only dilutes the samples, number two, one sample may be having inhibitory material that inhibits to other. And number three, one time came when almost 50% of our specimen were positive. So if you have to pool of 10 and you get it positive, you have to repeat, and everything has to be repeated. So it was not cost effective at that time when we had high prevalence. So we started with less than 1%, then 10%, then 54%. And Alhamdulillah, now in my lab, we have come to 3-4% positivity. So it has gone down with that. So size of the pool, repeat testing, some people actually, but in Germany, actually, they did this pooling for the mass screening. And you can do 100 tests to test the 1,000 people if you have got the pooling. And Germans have initially actually pooled the specimens and then tested whole of the population. I don't go in that detail because we have not done it, and I don't. So what is the primer? Primer is the short fragment of the DNA, which is complementary to the transcribed viral RNA. What is the transcribed viral RNA? You extracted the viral RNA, converted it into DNA by reverse transcription, and then you actually separated two strands. And primer actually is the complementary to that. It can attach to one of the strands. And these primers, actually, they are attached, and we put the, against it, we put certain bases, and then the genome is amplified, and then we can detect the virus by having the similar probes which are attached with fluorescein dyes. And as the probe attaches to its target, that dye is actually released. So that mixture is placed in the reverse transcriptase PCR machine, and actually there are cycles of cooling and heating. The strands are separated, then closed, then separated. And very interestingly, one gene can be transcribed into millions or billions. That's very important. So this is the test which multiplies exponentially. Antigen doesn't multiply, antibody doesn't multiply, but this test actually they multiply. 2 copies make 4, 4 make 8. And then at the end of 35 cycles, 35 billion new copies are produced from one copy. That's very important. So there are certain people who even go beyond 35 cycles and they go up to 40, 45. I don't believe in that. That virologist is very important to tell at what cycle you should report it positive, at what cycle you should repeat it when it is between 30 to 35, at what cycle you will call it negative. So that's very important. But I go maximum to 35 cycles. Beyond that is a non-specific. If you've been positive, then it might be false positive testing. So this is one gene multiplying into so many. So I don't go in detail, but and I'll just say it is highly sensitive and specific. And it is completed in 3 hours. That's very, very important. Virus isolation used to take about 10 days. Significantly faster, and there is low potential of contamination because it's closed tube. It is most accurate test and cannot detect past infection. It just detects when the virus is there. So for past infection, you have to go for the antibodies if you want to know. And then it detects the small amounts of viral DNA, and we have to differentiate the positive signals. It might not be due to the other coronaviruses, which we can give it false positives. Targets which we have to select for the primers, they have to be very specific in that. And then large capacity. The main thing in this country was, when the testing started, was the capacity. In Chuktai lab, I could easily enhance my capacity to 5,000 tests a day when the whole country was not doing 2,000, because we are the virologists and we are prepared and we could actually expand ourselves. So having a big, large capacity means that you have to have planning and disaster management. So having said this thing, initially they were targeting the test as S gene, but later on they found this reverse RNA-dependent RNA polymerase helicase beta gene, then N, then E, and slowly and gradually the WHO asked us that you use this gene and one of these genes. So what we are now doing, we are doing it in 3 or 4 genes at the moment. Initially, we used to start with 1 gene. If it is positive, we were repeating to know whether it is specific or not. But now we are doing about 4 genes at one time. And as far as the DNA extraction is concerned, there are 2 methods, 1 step and 2 steps. 1 step is very quick, and we are doing it. In 45 minutes, you can extract it. 2 steps is a longer method, but 2 step is needed for the genome sequencing, for research studies. 1 step is for diagnostic and 2 for the research studies. So that is, we can archive the sample then. So I just say that timing of the specimen taking is very important. It should not be too early, because one person is exposed and they come, अगले दिन आ जाते हैं कि जी मेरा टेस्ट करें, और वो इन्फेक्शन अपनी उनकी खत्म हो चुकी होती है तो फिर आते हैं. That's very important. It should be taken when person is actually having an ongoing infection. Limit of detection due to late infection with atypical manifesting is important. People say जी आज मेरा पॉजिटिव है, 2 दिन बाद नेगेटिव क्यों हो गया? So that's a ridiculous question. Quality of sampling is important. I've already mentioned about bronchoalveolar lavage, then quality of testing, then quality standards. I don't suggest the antigen testing, which is actually it is sensitive 38% maximum, 15 to 38%. And that is actually doesn't help us much. We used to do antigen testing for influenza, for respiratory syncytial virus. But in this case, the sensitivity is so low that it cannot be relied upon. So as I have already mentioned about S, N, and E proteins and other things, antibodies I leave to my next colleague. And as I mentioned, there is not only 1 coronavirus, these are 7 coronaviruses. And we had a very bad experience with the MERS-CoV in Middle East. There was a SARS one, and they are there. And this is just, I'm showing the Governor of Punjab visiting my lab just to see and just to visit. So what actually is very important is the timing of the sampling, proper extraction, and then proper testing with good quality. We are covered by CAP, College of American Pathologists, as far as our quality is concerned. Also by NIH. Capacity, as I mentioned, is very important. Our capacity should be extremely important. And then, as I mentioned, that if we do the research, we can resort to the gene sequencing. By gene sequencing, we can understand which strain is there. Because if there is a variation of the strains, by sequencing, we can pick that differences. And that can tell you which kind of strain is moving in that area. But in Pakistan, still, I think there is not much strain difference, although we have received strains from different countries. And still the virus is behaving the same. Maybe that in future, there are so much variation that this virus may be pacified. But as this is the first pandemic of a coronavirus so far, we have seen the pandemic with influenza A. This is the first time. And influenza A, we know that after the pandemic, then the virus becomes less virulent and it becomes easy disease, because when in 2009 the swine flu came, it was very dangerous, but in 1 or 2 years it became quite mild. In 2009, 284,000 people died in the world, but now not so many people don't die with that. So whether this virus becomes mild or disappears, we don't know about that. When this virus will disappear, we can predict in Pakistan, but not in the world. It is going on in the different parts of the world. Whether the herd immunity will be long lasting or not, I don't know exactly about that. There are so many questions. This is a new virus, and the viral diagnosis is very important to diagnose individual case, to know where the disease is going, to know about the variations, and then to see when the person is infectious, when it's not infectious. But one question, I will just say you one thing very important, that in some cases, the person remained positive for 5 weeks or 6 weeks. But it does not mean that person is infectious also. Because when the cells are broken, flecks of cells remain inside the nasopharynx and the RNA is there. And when you detect the RNA, it doesn't mean the live virus is there or not. So WHO says that anybody who remains symptom-free for 4 days is no more infectious, is no more needed to be isolated, provided he has started infection 10 days early. So 10 days, if a person is asymptomatic and detected positive after 10 days, there is no need for isolation. Even if become sick after 3 to 4 days, is no more infectious if person is totally asymptomatic. So retesting a person and retesting, I do not suggest. Even if a person remains positive, it doesn't matter much. Clinical diagnosis is more important than that. So, ladies and gentlemen, with this, I just finish because I was just about 20 minutes. And then, inshallah, we will come back in the question answer session. Thank you, sir.
Thanks very much, Dr. Zaman Tariq. I'm sure by this elegant explanation, many of the questions would have been answered automatically, but still if there are questions remaining, the viewers will send us through the Facebook comments, and inshallah at the end, after the second speaker, we will take the questions. And it is another privilege to welcome Professor Najeeb Ul Haq. Professor Najeeb Ul Haq is the clinician of high eminence, principal and dean of Peshawar Medical College. Prior to that, he's graduate served on medicine there for a long time. He is fellow of Royal College of Physicians and fellow of College of Physicians and Surgeons, Pakistan, and extensive experience. He has been involved recently in the community, and I think at this time he is the very appropriate leader in the medical community who we have invited to talk on the serology and antibody testing for COVID-19. I request Professor Najeeb Ul Haq to start his presentation.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. I'm going to talk about COVID-19 antibodies testing, and I'll be touching more on the practical aspects today rather than going into the detail of the structures and basic immunology, which partly has already been covered by Waheed, and I'll be trying to more practically involved as a clinician, so I'll be trying to talk more practically about the antibody testing. You have already seen this picture. We know there are different antigens on the virus, and that's why it is called coronavirus, because it's like a thorn that you can see. जैसे ताज से मुताल्लिक है ये कोरोना वायरस कहते भी इसीलिए इसको हैं. And there are multiple proteins on the surface and inside the virus. And before going into the antibody, I'll briefly tell you why body produces antibodies is basically telling us about our immunological response, how body reacts to a virus, outside virus. Let me give you the example of hepatitis B, which is very commonly known to almost all of us. And if you look at this picture, there is something called HBsAg, which we normally test. It's a protein. Then inside are the DNA, then single and double stranded. Then inside further are the core antigens and the E antigen that you can see. And if you look at this picture, every antigen, against every antigen, body can produce an antibody. Like against hepatitis B surface antigen, it can produce an antibody which is called anti-HBsAg antibody, or simply known as hepatitis B surface antibody. Then again, the core antigens, it can produce IgM and IgG antibodies. And again, the E antigen against the E antigen produces anti-HBe. Why did I show you this picture is because there are certain antibodies which are protective, which give you lifelong immunity, while other antibodies might not have any role in protection. Like if somebody has got a surface antibody positive, we know if it's more than 10, it gives you lifelong protection. But the others don't give you protection, whether it is IgG, IgM, whether it is HBe antigen, antibody, or core antibody. The only thing is that core IgM means active viral virus B present in the body. We do not know exactly how the coronavirus behaves, but we do know that exactly like this, there are multiple proteins in the virus against which antibodies can be produced. And this is how a coronavirus looks like. And there are multiple glycoproteins against it, M protein and other proteins on the surface and inside the virus. Now, this is important to know that the types of antibodies that presently have been isolated and we know of are the IgM, IgG, IgA, and other neutralizing antibodies from the spike proteins. Now, the commonly tested one that we have in practice here in Pakistan and most of the world is the IgM and the IgG. Now, sometimes they are done together in one test, and sometimes they are done in isolation. IgG is done separately, and IgM is done separately. But generally, when we say IgM, it means that this is an acute infection, and it tells us about the ongoing activity of the virus. Unfortunately, the IgM specificity and sensitivity is not much yet. And the commonly used tests that we have in the market now tests both IgG and the IgM together. We have the others one as well, but they are not as reliable as these ones. Now, when we say specificity of a test, it means that how specific a test if it is positive. Is there a chance of false positivity or not? So the more specific it is, the lesser will be the chances of false positivity. Well, sensitivity means that a test, what are the chances that it would miss a positive test? In very simple words, if I may explain it. Okay? Like some kits have a sensitivity of 85%. That would mean they would miss 15% of the positive tests. That is important to know because the kits that are available in the market, we should be asking the laboratory person that which kit is he using for the antibody test. The two most commonly available now which test both IgM and IgG together, in the one which is commonly used in Pakistan particularly, they are FDA approved and they have been given emergency approval, in fact. And the one we are using here commonly in our lab, in Al Khidmat lab, and other common labs, has got good sensitivities from Roche. Specificity is very good. They claim a sensitivity of even 99%, but the UK Public Health, in its independent analysis, confirmed that it at least 85% sensitive, which is still good. But the specific IgM and specific IgG test, their sensitivity and specificity are still questionable. So we cannot rely on that too much. Like in case of hepatitis B, I gave you the example, we can rely if hepatitis B core IgM is positive. I'm almost sure that this person has got acute hepatitis B. But in case of COVID, I may not be as sure. For that, as Waheed said, we will have to do another test, which is the gold standard at the moment, which is the PCR test. The unfortunate thing about the PCR is that many of the patients who test negative for PCR, they may still have COVID. It depends on the time of taking specimen, on the mode of specimen from where you have, whether it is only nasal swab, nasopharyngeal swab, sputum, bronchial secretions. Obviously, bronchial secretion is the most difficult one, except in cases who are on ventilator. And as he said, the number of cycles that you amplify it, the more will be. Many tests who initially become negative for PCR, they may be later on positive for PCR. Now, are these antibodies protective? That, as I already said, that is a very key question that will be answered only with time. But presently, the evidence is that reinfection in antibody positive subjects is very rare, almost not reported. There had been few cases where there has been doubt that they have got reinfection or they have got another mutant virus to which they got infected, but we still don't know exactly whether they were positive for PCR, whether they had, because the PCR would not 100% tell you that it's a living virus. As he said, it may still be a virus secretion, but they may be just dead virus. So at the moment, we have this evidence that those who are antibody positive have got very, very small chance of getting reinfection with the same virus. Then we are also hearing about the herd immunity. The herd immunity will only come when there is a protective effect of the antibody in the individuals. And that will lead to a lower transmission ultimately. The third evidence that we partly have is the SARS infection in 2003, and a study later on, 2 years later, conducted on some hospital healthcare workers showed that they still had antibodies present. This and the SARS virus belongs to the same family, and we hope that the code 2 will also have antibodies which will be hopefully protected. Keeping in view all this, there is a question whether we should start using this opportunity of presuming that it is antibody positive is protective antibody, or we just wait and see and let the virus grow further. So it has to be balanced, and we know now that people are using plasma as a treatment option. It is for the same reason that it is being used. So in nutshell, I would say that antibodies that we measure with common tests are protective. Multiple studies are going on, but unfortunately, some of the studies have shown that these antibodies will fall down very quickly. In some cases, within 3 months, the antibodies would be negative. But we still don't know exactly the behavior of the antibodies, what would they do in the future. We need, because that study is at a very small number of patients from China. We still need large studies of antibodies to see the level that goes up, and then it comes down for how long it stays. So that will tell us the exact position whether these antibodies are protective or not, and for how long they are protective. Because they may not be lifelong protection as in case of hepatitis B, but there may be some protection as in case of influenza vaccines that we take. Some people take, old people and those who are sick, they take in every season. So we don't know exactly, but we hope and wish that these will be protective antibodies which will lead not only herd immunity, but also give individuals the protection as well. Now, the other question is that are antibodies diagnostic of, number one, acute infection? As I said, can we equate IgM to PCR? The present answer is no. PCR is the one that has to be done to see whether acute infection are present. It has its own problem, but IgM is much lesser sensitive and specific than the PCR. The standard still is PCR, deciding about the treatment, particularly antivirals. Evidence of recent and past infection, COVID antibodies, yes, it's very good. We know that the antibodies would indicate that either the person has recently infected or had infection few months ago, but this is something that can tell us about the community prevalence. It may not be very important on the individual level, but it can tell you the overall prevalence of the risk of infection of a community. Like we recently conducted a survey of the healthcare workers in Peshawar, screening more than 1,000 healthcare workers. And it's still not published, submitted to the BMJ. We noted that 30.7% of the healthcare workers were positive for hepatitis B antibody, for COVID antibodies. It was a huge number. And almost 50% of them were asymptomatic. That means that they had COVID, they did not get any symptom, but they did develop antibody. And this is a dangerous situation, because those who are asymptomatic working in hospitals and they have COVID, they're transmitting it to the other colleagues, to the hospital staff, and even to their own families outside, but they're asymptomatic. So this is something which is quite worrying, and we are recommending for the same reason that there should be probably spot checks at different time intervals in the hospital workers. Is there a cross-reactivity? There is a possibility, yes. We don't know exactly where does it cross-react. It's just an anecdotal feeling that although Typhidot is not a test to be used, it should be banned, and probably it has been, but there is an IgM cross-reactivity of the Typhidot test. So we still don't know, but there is a cross-reactivity that we know of. But we should know that the false positive in the kits that are being used commonly is very rare, because the kits are very specific. That we should know. The other question is, how long will it take to develop vaccine? I don't know, but there is a chance, hopefully, that we will have vaccine. How does these people develop this vaccine? If you look at this figure, there is a protein here against which an antibody is produced, and there is, for example, another protein here, and again antibody is produced. If you take that vaccine, this protein, and put it here, now what would happen that this virus will start producing now this antibody as well. If you take some protein from a virus, put it into yeast or another thing, bacteria or a yeast, or then that can start producing the antibodies. It could be synthetically as well, but this is another method. And this is how the hepatitis B vaccine is produced, that they take this part of the surface antigen, and then they put it into yeast, and then if you look at this, these are the particles which are produced, ultimately purified, and then saved as a hepatitis B vaccine. Let's hope that inshallah, very soon, the vaccine will be there, and we'll be able to manage this. Alhamdulillah, in Pakistan, what we found, another interesting thing in our study, was that the social distancing we know of, and the risk factors going to the community, shopping malls, markets, wedding, etc., we found that going to the mosque was not a risk factor. And that was something strange for us, that it's the same as the other things are. But then probably it is the strict observation by the people who are in the mosque. They follow their ulama advice. They are always have to wash their hands and face 5 times a day. The prayer area is usually clean. So probably the personal and the environmental hygiene that is protecting us in the mosque and giving us less risk compared to going to the markets and going to the shopping malls. Let's hope that we soon have vaccine as well, inshallah, and can use that for preventing the further cases of COVID. Thank you very much.
Thank you very much, Professor Najeeb Ul Haq. We have some good time for question and answers. My first question is to Dr. Waheed Zaman. This is asked by Dr. Danish Sharif. He's asking that is it important for the clinician to know which method the lab is using for PCR testing, or all the labs have got the same method? Please unmute yourself, please.
Yes.
Thank you. Actually, if a virologist is actually leading the lab, he should not put this responsibility to the clinician to know which method is being done or which is not being done, number one. Because it is our responsibility, not their responsibility. We should choose the best possible method, number one. Number two, we should have a good internal quality control and good external quality control. So if the individual physician, they start deciding about the types of the test, and they start doubting in which lab and that lab, then it doesn't work. But whatever I say at the moment, there are certain labs which have got good quality, others might not be having that standard quality. But what we said to the government at this period of time, that please allow everybody to do the test, whosoever can do the test. So what I'm saying, that at the moment we are doing the TACMAN antibody TACMAN PCR test, and TACMAN PCR test is actually FDA approved. We also do the Roche test, which is also FDA. We also did some Abbott test, which is very slow methodology, which is also FDA approved. But what actually is done is that NIH Pakistan has established a quality control system that they are sending the specimens to all different labs, and they are actually watching their quality of different labs. And as far as we are concerned, we are also getting it by College of American Pathologists. So it is our job to ensure the quality, and then the clinician should have a trust in the lab if the lab is properly supervised. That's very, very important. And where the lab is not properly supervised, lab must have the kind of proficiency testing. Proficiency testing means that they should send some sample to me or to NIH, or get my sample and then compare the results for that. So that is what I'm saying. So rest assured, it is not actually the quality of test, it is the quality of the sample. And when the sample is being taken, that's very important. And number two is very important, that people start comparing different labs. And when they start comparing different labs, आज वो मुझे सैंपल भेजेंगे, 4 दिन के बाद शौकत खान में भेजेंगे या किसी आगा खान में भेजेंगे, और 4 कहेंगे जी आपका पॉजिटिव टेस्ट का नेगेटिव हो गया, इसका नेगेटिव. So they should not take this responsibility on themselves. They should put the responsibility to the pathologist, whosoever is there. So I assure you that in the good labs, all good labs, I'm not saying my lab, even at the government level, we are now doing the test on the FDA approved kits, which are well under quality control. So take care of the specimen taking and proper timing when the specimen is going to be taken. That's more important than that.
Thank you. Professor Najeeb Saab, the question is that if a person has got symptoms suggestive of COVID, but his PCR is negative, he's admitted for his clinical picture, is there a value of doing antibody testing on that patient in the hospital?
Yes, that is where the antibodies role come actually. Many patients, almost 30% would be PCR negative. Although the type of PCR now is improving and we may have less and less false negative, but that is where the role of antibodies come. So if somebody has got symptoms typical of COVID, irrespective of what the PCR says, we should treat him as a COVID positive patient in terms of symptomatic relief, not in terms of giving him antivirals. But if he has got a positive antibodies in a negative PCR, we should take it as a COVID positive patient. My next, the other issue that was about the clinician, I fully agree with what Waheed said, that we should trust our laboratories. But unfortunately, many of the labs do not even write which method they are using. So that is the area where it becomes doubtful that which method somebody has used, especially for antibodies. The NIH has got a very good system. Even when we started PCR in Peshawar Medical College, they had visited us twice, and then they made sure that we send them the samples and the quality control is there. But in antibodies, unfortunately, there are many issues. And as he said, that once PCR is positive, that's it. We don't need to repeat it somebody from somewhere else. If it is positive, it is positive. If it is negative, then one might consider repeating it after 2 or 3 days or whatever the symptoms tell us. But generally speaking, if PCR is positive, it's positive. Once positive means positive.
Professor Waheed, do you have any comments on this?
So thank you very much. Actually, he has covered it very nicely. But problem arises when I give the positive. Initially, the people were actually taken on isolation by the government of Punjab. And then the people used to have fake reports in their hands. And they used to show on the press. So that was a time when it was more of a political thing rather than of any clinical thing, number one. And number two, nowadays we have got the contract with about 5 airlines of the UAE, Etihad, Emirates. And if God forbid someone gets positive test, we have got the contract with the airline, we have to tell them that he is the positive. Rather than accepting it positive, they will try to bring from somewhere a negative result and then starting making a fuss with the airline and with us. So what actually I agree with Dr. Najeeb Ul Haq, when it's positive, it should be positive. But why the people challenge, it is not due to clinical reason. There are some other reasons where the people have been manipulating and crying here and there. And the reason is obvious, that if someone wants to travel to Canada, and then they want to have negative in any way. So that's where the problem does arise. But as far as the, yes.
Now that by the grace of Almighty, the disease is getting tamed down in Pakistan, we, the clinicians, are asking ourselves and to you, the experts, that is it herd immunity? Is it the virus is getting tamed by its virality is less? Or is it the protective measures by the public which is making a difference? What do you think?
I am allowed, sir. I heard from Dr. Najeeb Ul Haq very nicely saying 37% of his staff, medical staff, was antibody positive. Here, up to 60% of the medical and paramedicals were found positive by me. And Chuktai Lab has actually given a free test to all the medical and paramedical staff. Whosoever can show his ID card that he is a doctor or a paramedical staff can get free test of antibody done at the moment. So what we have done, we have found even up to 50 to 60%. But it was a wrong way of getting the herd immunity. Herd immunity I have heard with the vaccine. Better to get it with the vaccine. But I think about 50 to 60% when people have been exposed. Even in Lahore at one time, I have estimated on 15th of May that about 7 lakh people were infected. On 6th of June, we thought that is about 2 million people were infected at one time, actively infected at that time. So what we have seen, I think 60-70% population has already got it. And number two, now the people have also adopted good measures. Initially, they were not listening to us. But with the gradual passage of time, when they have seen the people dying in their families, then, what they did in the Eid al-Fitr was very bad. But in Eid al-Zuha, there is a general tendency of keeping a distance and saving themselves. So I think it is not one thing, there were so many other things which have acted together. And when the virus is going down, it means that I have myself seen, as I mentioned, that I have seen positivity ratio in the tested 54%. Now I'm seeing 3 to 4%, even less than that. So what has happened? It has happened actually gradually. And I think we have gone through that. We have tasted the virus and we have gone through. And luckily, 80% of the people remain completely asymptomatic with this virus. That's a very important point, which must be noted. That is the reason that people have gone through this virus without knowing this thing. So herd immunity has definitely worked, and still, I will advise that it doesn't mean that we should stop taking the precautions. They will remain there. We are opening the schools on 15th of September. I have told to the, on behalf of the government of Punjab, to the Green Line and Metro, open it in October. And inshallah, by the end of September or early October, we will be totally out of that, which I am predicting, inshallah. That is on the basis of.
Dr. Waheed, although you and Professor Najeeb has touched this a little, but again, some of the doctors are asking this question. What are the chances of for a person to get infected second time from COVID disease?
Good. It's very good question. Actually, there were few doctors, one from your hospital, which they said initially it is a reinfection. You have heard about that. It's a very popular case. A person got it, he was positive, and then he became asymptomatic, test was negative, and then after. But all the American studies which they have done, they have followed hundreds of thousands of people who have been infected. They say it is not the reinfection. It may be reactivation. Reactivation does occur. The virus remains in the body for weeks and weeks in some cases. In immunocompromised people, yes, of course, it can remain there. And there are some other people in which the virus can be reactivated. So there is a difference between reactivation and reinfection. So far, there is no solid evidence, as far as the American studies are concerned, that there had been a reinfection. Although there are all reports throughout the world, Japanese said they were, Chinese said they were reinfection, we said, but I'm still convinced that this is one-time infection and it doesn't affect you second time, inshallah.
Thank you. Professor Najeeb Sahab, you mentioned that the getting so many positive health professionals, like in the order of, like you say, 30%, Dr. Waheed says like 50 to 60%. And you raise this issue that these healthcare workers may be a risk to other patients in their families. But on the other side, it could be a good news that they are already immune and the danger from them is minimized to the patients. So how would you compare these two aspects of same finding?
Maybe I couldn't explain it properly. That's why that question raised. In fact, it was that window period where they got infected, they were asymptomatic, they were having circulating virus in their body, they were excreting virus in their respiratory secretions, but they were asymptomatic. That is the time where they are infective to others. But once they develop antibodies, then we know they are no more infective. So it's primarily that period where they are asymptomatic, but they have active viremia present in their body, where they are a threat to others, and including their families and including other and their colleagues. Because if 50% are asymptomatic and they are carrying virus, say for 2 weeks or 1 week even, that 1 week is the dangerous one where they are infective to others. But once they develop antibodies, then they are no more infective. What we saw was that the doctors or the nurses or the paramedics who had antibodies, but they never had symptoms. That means there was some period in the recent past where they had the viremia, but they were not having symptoms, so they were a threat to others. That is the problem. The other is the difference between the healthcare workers. We had a healthcare worker survey across the board. It was a cross-sectional study. Whether somebody was symptomatic or asymptomatic, people when go to the lab, usually they have a reason to go to the lab even with mild symptoms. So that's why what Waheed said might have higher level of positivity comparing to what we saw. But whatever, it's still a very high level that we are getting. Antibodies means that we had recently been infected with the virus and we have overcome the virus. But that, in other words, means that we are at a, me or anyone else who is not antibody positive is at a higher risk. So it's the initial phase where they are infective, when they are a threat to others, but when they develop antibodies, obviously they are not.
Dr. Waheed, Dr. Iqbal Khan is asking that there are some suggestions, particularly coming from the West. There is not much evidence that the if protective antibodies are there, they are getting vanished over a period of time, and maybe after a few months, patients will be again vulnerable. The question is it a real threat, or it just maybe a kind of thought by the vaccine makers to get their market open in the future?
Thank you very much. Actually, what Dr. Najeeb has very ably told about antibodies, and he has very nicely mentioned that these antibodies, they do not correlate with the immunity. They are not neutralizing antibodies. Neutralizing antibody testing is a very rare phenomena which the vaccine producers are doing on cell culture, and 35 cases or 40 cases. So, in the absence of neutralizing antibodies, we can't say whether the person has got neutralizing antibodies or not, number one. Number two, when SARS-1 came, as Dr. Najeeb Ul Haq was mentioning in 2003 in China, they found the people whom they tested, 100% of them, they were positive after 1 year with antibodies. After 2 years, 20% of them, they lost antibodies. After 3 years, 50% of them, they lost antibodies. With this virus, we are still with few months. In this country, in Pakistan, in Punjab, if you say, or in Khyber Pakhtunkhwa, the virus came in the end of March. And how many months have passed? In 3 or 4 months, they are so insufficient to make these kind of deductions. We should believe in one thing, that the antibody which we are testing, we are not still testing with the neutralizing antibodies. And as far as the vaccine production is concerned, I tell you honestly speaking, in case of SARS-1 and in case of MERS-CoV, vaccine attempts were made, but the viruses disappeared before the vaccine came. Still, the British people, they say that vaccine will not be available till the end of this year. Even if they get by the end of this year, then there will be process of the legislation and their approval, and then mass production. How many months after it will come to Pakistan after mass production? And that nation which doesn't accept even the polio vaccine and measles vaccine, how will it accept it? There are so many other questions. So I do not agree with Prime Minister or anybody, vaccine aayegi to mamla theek ho jayega. I say, inshallah, the virus will disappear before the vaccine comes. I'm quite positive about that. And even with hepatitis B, vaccine has definitely helped us. But my teachers have told in England that virus started going down in prevalence much before the vaccine came. So vaccine is a complementary. It is not actually the answer.
This remains a pleasure and for me a privilege to thank both these speakers, very experienced clinicians and pathologists, and I thank them very much on behalf of Pakistan Islamic Medical Association viewers. And inshallah, we will be back with our viewers next Sunday with another topic, which is the other tests which are involved in the COVID management and also the radiology part. Thanks very much. Allah Hafiz.
Thank you, sir.